Thank you for having me. It's really exciting to be on the West Coast while I'm on the East Coast. And what I was asked to talk about tonight is a program that we call the Comb Project, although you'll come to realize it has nothing to do with building comb or anything like that. It's just an acronym for conventional and organic management of bees. And so to repeat my intro a little bit, I added a few slides here to my talk to show um, my background so you can get to know me a little bit. Uh, as Etienne said, I learned to love bees from Dewey Caron way back in the mid 1990s at the University of Delaware, and Dewey and I have kept in touch ever since. And here we are at the Eastern Apiculture Society in 2017 when they hosted it in Delaware. I was lucky enough to go to that and see him there. Um, I then went on to do my PhD at the University of Manitoba. And early on, after I graduated, I tried Googling myself, and this is the photo that I found. And funny enough, it's still online. What I was studying there was using formic acid on indoor wintered bees. And so I had to wear this crazy getup that would put fresh air into my hood while I went into the formic acid laden air. So this is a really funny picture to have online. But apparently beekeepers there are starting to use this, or have been using this as a treatment. I then taught biology at Kutztown University, and I did research, 100% research appointment at Penn State, and now I'm 100% extension. So I have to say I have finally landed my dream job, which is, just talking about bees and talking to you guys and writing and doing everything just about bees all the time with no other requirements. So it's really fun. Extension is an amazing organization that brings research to, quote, farmers, which I consider beekeepers to be farmers of bees. So I don't think I need to tell you about all the different stressors that honeybees are under. Many of these in this diagram are things that are, for me, out of my control. I ask a farmer if I can put bees on their land, and the bees fly wherever. I can't really control what pesticides other people are putting onto their properties. I can do my best to keep the bees safe from pathogens like viruses and bacteria. But what I can really do is control my beekeeping practices. So that's something that I'm passionate about, doing applied research that's applicable to you immediately. And I can see the results right away as well. So one thing that we talk about a lot, but maybe aren't properly defining, is people's beekeeping philosophy. So in life, you may have a philosophy about, like your lifestyle, how you think, what you eat, things like that. In beekeeping, you can have a philosophy about what your role is as the beekeeper. So we can categorize people based on their philosophies, and your philosophy is a guiding force for what we call your management system. So this figure right here is a diagram from a paper that we submitted where we were actually studying beekeepers, not bees. And we were able to purchase data from the Bee-Informed Partnership surveys. And on it, you're asked, like if you do the management survey, the extended long management survey, at least back in the 16-17 winter, you could answer a question. It was like question number 80, like deep into the survey, about whether you're willing to use any treatments for mites, or you only use natural chemicals, or you will never use any chemicals in your hive, right? And so what was happening during the management survey really is Bee-Informed Partnership was asking you your management system. You may not have written it down though, and you may not realize what your system is. So this diagram can be used for that, and it can be used for new beekeepers to help them learn where they fit in, right? So a new beekeeper is learning how to keep bees, and they come in probably thinking there's one way, and then they quickly learn that there's, many, many different ways and many different decisions to make along the way. So this diagram is showing you the degree of intervention from low to high of all these different things that we're deciding. So if we look at just the first line, we have, what do you do in terms of chemical treatments for varroa mites? Well, if you do nothing, that you're not intervening, so that's very low intervention. If you use essential oils or acids, that's kind of in the middle. And if you use synthetics, that's way at the end of high. In my opinion, I chose high for that because not only is it a long-term treatment, it's a 42 days of amitraz and then 2 more weeks before you can put your supers on. That's a really long time. But also because when you remove those strips, you're not really actually removing the amitraz per se, because some of it has gotten absorbed into the wax. Then we go down and we can look at, cultural controls that we may be using for mites, for example. If you're not using any of the available cultural controls, low intervention. Screen bottom board is pretty mild. It's just the mites are falling through. It's a little bit more intervening if you're causing a brood break on purpose. And then here on the far right, we have using small cell comb, which is high intervention because it's fundamentally changing the bees. So anyway, I'm not going to go through this entire diagram, but you could sit down, you could print this out. By the way, at the bottom here is how you can find this paper. It's free to download for everyone. There's no paywall. And this diagram or this figure is also free to use. So you're welcome to print it and use it as you wish. So you could have a room full of people and have this printed out and ask them, what do they do, right? So they would go through here and they would circle, okay, I'm comfortable with, formic acid and thymol. I wouldn't really replace the queen on purpose, only if I need to. I like to use sugar syrup with some kind of additive. Why am I keeping bees? Do I want to produce honey and extra bees, or am I just pollinating or whatever? So you go down the line and you circle all these things, and then in front of you, you have your management system. This is your approach to beekeeping. This is your philosophy embedded into this. So we looked at the survey respondents' management systems, and based on that question number 80, if the respondent said they were willing to use synthetic chemicals, we called them conventional. If they said they don't use conventionals, but they still use chemicals, like formic acid, we considered them to be organic. And if they don't use chemicals at all, we called them chemical free. So you can see here that these are sort of color coded, and that's what you're going to see in the future diagrams. So we have the data from many, many different questions, all summarized in these figures like this one, which you're going to see dots appear on the screen. And these dots are going to be color coded by like all natural, organic, and conventional. And in this particular diagram, we're looking at what are the beekeepers using these management systems or these philosophies feeding to their bees. Okay, so here's all the different dots. And what you can see is that the management system, the decisions that people make are not random. They really fit into their philosophy. So conventional beekeepers are feeding whatever carbohydrates they can. It could be high fructose corn syrup, could have additives, could be whatever, and they're feeding protein. We have organic beekeepers who aren't really giving them pollen patties, kind of like out of sync with nature, and they are giving them some of the carbohydrates, whereas all natural beekeepers definitely aren't feeding pollen patties and are more likely to be feeding them actual honey or leaving behind a ton of honey. So now we're going to look at what are the goals of these beekeepers, color-coded in the same way. So now you can see, again, they kind of break out into groups. And the people doing conventional beekeeping management systems are keeping bees for a reason. They want to make honey, and they want to make money, and they want to do that selling the honey and selling bees. People all the way on the right here with the natural beekeeping, not interested in harvesting honey, really just keeping their bees for teaching or pollination of their own garden or research. So you can see where your philosophy and your goals, they kind of go together and influence your management. We also looked at the size of the operation. So this is totally the same dots, only this time color-coded by operation size. So backyard or hobbyist beekeepers have less than 50 colonies. Sideliners are up to 500. and commercial is over 500. So I'm sure you can imagine that the scale of your operation is going to impact your management. So again, what we see is a nice tight circle for commercial. You know, if you have thousands of colonies, you can't really think about each individual one, and you want to blanket react, right? So they're very often just for putting in the synthetic chemicals, probably prophylactically treating for Nozema. People in the middle are kind of doing almost the same thing, but not quite the same as the backyard beekeepers. So what I find the most fascinating about this whole paper is the broad range of things that backyard beekeepers are doing, right? Some of them are doing things very similar to a commercial beekeeper, right? Why would they need to act like a commercial beekeeper? Probably because their mentor is telling them you must do this or you should do that. So this is what the people that I talk to, right? So I'm from Pennsylvania. 98% of Pennsylvania beekeepers are hobbyists. There's a small 1.5%, you know, that are sideliners, and there's like 3 guys that are commercial, right? That maybe take their bees to almonds. Everybody else is kind of stays local. And like I said, 98% have bees in their backyard, or maybe up to 50 colonies in their backyard and several other people's backyards. So that's really who my stakeholders are. That's who I am interested in teaching how to keep better bees. So when I go to meetings of beekeeping clubs, I can see these 3 sets of people in the room, and often they're fighting. Or the chemical-free people are completely absent because they've been totally alienated. Everyone keeps telling them to treat. They don't want to treat, and they don't want to be told to treat again. And so if you think about the philosophy behind what each of the groups is doing, everybody thinks they're doing what is best for their bees. So a chemical-free, treatment-free beekeeper thinks that bees should be treated in an all-natural way, and that's what's best for the bees. They know what they're doing. And a commercial or conventional beekeeper is like, no, you really have to treat. You have to do these things. You have to manage them. And everybody thinks they're doing what's best. But what I wanted to do is set out and get the data, right? Show me the data that says, how healthy are these bees? How well are they doing? How much honey are they producing side by side? Well, there's no beekeeper that does multiple philosophies, right? Because that beekeeper has their own philosophy and they're treating all of their bees the same. So as a researcher, I'm like, okay, I need to do this. I need to set out and get the data. So we did a literal side-by-side comparison of 3 management systems. So each apiary had representatives from the conventional system, the organic system, and the chemical-free system. Now keep in mind that we treated these bees like we were sideliners trying to make money through honey and bee sales, okay? We split hives to get our numbers back to where they were, and if we had extra, we sold them. And also we were always trying to make honey with these stationary colonies. So for our research, we had the 3 philosophies. So conventional management includes all of the cultural controls, all the organic acids and essential oils, but also things like amitraz. Organic just cuts out all the synthetics, and chemical-free is where you don't treat at all. So these are the 3 systems that we compared side by side. And to determine the actual management system down to the what kind of bottom board should we have, what should we feed them in winter, all those details we got from beekeepers. So here we have, all in the same room, 30 beekeepers with all different management systems. And we sat them down by philosophy and we said, okay, you need to tell us all the answers to this whole list of questions, right? Like, should we get standard comb or small cell? Should we treat with oxalic or formic or thymol or amitraz? And when should we do that? What do you feed them over the winter when they don't have enough food? On and on. We sat together for a whole entire day. And I was very proud to have gotten these people all into the same room. They did not fight. But it was clear, even when you set 6 or 8 conventional beekeepers in the same table, they had to compromise on what fit into their management system. So clearly it's not as simple as these three, and there's a spectrum within each of the three. But we hammered out, with their help, what do beekeepers in Pennsylvania actually do? That's what we want to do based on these three philosophies. So there's a lot of different details that are different between these three management systems. So I'm gonna just try to spend less than 5 minutes telling them to you. So remember, there's conventional, organic, and chemical free. For chemical free, I'm just gonna describe the whole management system. So for chemical free, the bees came as packages from the fat bee man because he keeps small cell bees, and I wanted to have small cell bees on small cell comb, and I thought they would build it best. Okay, so we gave them 4.9 millimeter plastic foundation. The stakeholders told us they preferred a solid bottom board. The foundation that we gave them had 1 inch cut off of each side so that they had a choice and could build drone comb if they wanted. The insides of all the boxes were scratched up, and we used a duck cloth cover, and that was to promote the propolis envelope. And no treatments were ever given, ever. Although we did monitor mites, so you get to see how that went. In the winter, if they used up all their winter food, we would give them dry sugar, but that was extremely rare. So for organic and conventional, a lot of things were similar. They got all standard worker comb. Conventional chose a screen bottom board, where organic chose solid. Conventional used all standard comb. You know, you want as many workers as possible to make lots of honey. Whereas for organic, in the brood chamber, which was 3 mediums or the equivalent of 2 deeps, 1 in 10 frames was a drone frame. So we did drone brood removal for mite reduction. And we used a normal wooden inner cover with a notch for an upper entrance year round. And when these bees arrived from a package company in Georgia, we did an oxalic acid dribble. I was very happy to say that hardly any mites fell at all. So they were clean on arrival. For these in the first year, so they were on all new equipment, all new foundation. They had to build all of that out the first year. The mite levels never got above our threshold of 1% throughout the summer. But in the fall, we blanket treated. So every conventional colony got amitraz treatment. Every organic colony got a formic pro treatment. And then they got like a winter cleanup oxalic acid vapor. We then put a 15-pound candy board on the conventional, and we put dry sugar on newspaper, you know, mountain camp method on organic. Then we also followed them for year 2 and year 3. So you know that mature colonies have different problems. So we had to think about treatment more often. So in the spring, which for us is April, May, if the varroa mite levels were over 1% in an alcohol wash, we would use oxalic acid vapor. If we get to June and July, we would then switch over to formic. If they needed a second formic because they were still over threshold later in the summer, that was okay, but never a third, right? And so we were always trying to rotate chemicals using an integrated pest management approach. We were always treating an apiary based on alcohol washes until the fall. Then we blanket treated conventional with amitraz, and organic ended up getting formic if they hadn't had formic yet. If they had, then they got Apigard, which is our thymol. So I hope this isn't too confusing, but in a nutshell, conventional got treated at a 1% threshold and blanket treated with amitraz every single fall. Organic, always rotating oxalic acid, formic acid, and thymol throughout the years. Chemical free, no chemicals ever. Okay. These were placed on farms in both Pennsylvania and West Virginia. This is the site of the, I think, famous Appalachian beekeeping collective, where they teach out of work coal miners how to become beekeepers. And that year they were setting up 1,000 new colonies on new equipment. So Perry Keatsman called and said, hey, I just heard about your project, and what if I do some replicates using your protocol? So that was awesome. So we ended up having 8 different farms. Each farm had 3 apiaries, and each apiary was broken down into the 3 management systems. So this is our perfect ideal 2018 setup. So we had 4 chemical-free colonies, 4 conventional, and 4 organic, all of them inside of a bear fence, because every single farmer said, yes, there's bears around here. And you'll notice here that the order of the 3 varies. So it just so happens that 2 farms make up all of the 6 possible combinations of chemical-free, conventional, organic. So we made sure to do that because we didn't want drifting to make any particular treatment more strong than the other because they're on the end every time or anything like that. I also, a lot of people think we should have had 1 apiary chemical-free, 1 apiary organic, but I feel very strongly that microclimate matters so much, and you can have an apiary where just bees don't ever survive there. And if that had just happened to be 1 treatment, it would have had nothing to do with the management, would have had everything to do with something like wind. So that's why we felt very strongly we had to have them all in the same apiaries. So as I said, they arrived as packages. I considered those packages to be my comb builders because it was all new equipment and they needed to build up the comb so that they could actually make it through the following winter. I was already thinking about winter in April. And you'll notice that they had different genetics, of course. But what we decided to do about that is we tried to get the best possible queens we could for our area. So this is Devin Paderowski. He keeps bees in the Poconos area of northeastern Pennsylvania, and he breeds, Pennsylvania mutts. And what they are is they're hardy Pennsylvania bees that have overwintered here a long time, and they have some mite resistance. So the colony that he grafted from for all of our queens, for all of those colonies, which there was 288, they were all sister queens from a colony that lived in the soffit of a house for 4 years, totally untreated. He did a cutout and put them into a regular hive, and 3 more years without treatment. So to us, these were, if you believe there can be these, quote, treatment-free bees. They were local to Pennsylvania, open-mated though. So they were sisters, open-mated, and every colony got requeened. We really felt strongly that the genetics needed to be the same because we did not want to see differences based on and wonder if it was because of their genetics or management, right? We only wanted to see management. We did alcohol washes every month, basically April through October, each of the 3 years that we were following these colonies. Every fall, we took a sample of bees. This is after treatments had occurred, so October time. We took bees and put them on dry ice for lab testing, which included 3 different immune genes, Vitiligenin, which is a level of nutrition. We looked for Nozema, both species, and we looked for deformed wing virus in those bees. We wanted to see the health of the bees going into winter. We also collected honey production data on every colony throughout those 3 years. So we have 3 years of honey data. And of course, overwintering success was really important. So of all the colonies that went into the winter in October, how many came out in April the following year? So let's look at some results. Here we have honey production. This is averaged over the 3 years, and you can see that it's per apiary. And there was statistically more honey made in the conventional and organic systems. And you'll see it's clear, basically why, but 102 to 119% more honey produced in those 2 management systems than in a chemical-free system. Throughout this time, remember we were treating when we hit a 1% threshold. And so that was on an apiary by apiary basis. So I can't really tell you how often, we treated in general, but we did not have any trouble keeping the mites down when we did have treatments available. So you can see here, these are kind of tracking together. August was always when we treated, so the mites usually go down by October. Then this measurement here in April is, any of the colonies that were still alive got their varroa mites checked. So in orange, you can see what happens when you do not use any chemical treatments. Here in the first winter, the chemical-free colonies averaged about 4% mites, which I thought wouldn't be so bad. Got much worse in the second year and the third year. The mites were quite high each fall. And so this is sort of your snapshot. Everything from here on is your snapshot of October. What were the bees going into winter with? Okay, so the bees in the chemical-free system were clearly going into winter with a lot more mites than the ones that had treatments available. They also were going into winter then with more deformed wing virus. So this is the actual quantity of virus in their bodies. So without any treatments and without treating mites, you are also maintaining these deformed wing virus levels. In terms of Nozema, this is a little bit strange. We're not sure how our management systems had an impact on Nozema, but you can see that the chemical-free and organic were significantly different from each other, with organic having less, but conventional was kind of in between, not different from organic, not different from chemical-free. So this is Nozema cerani, which by the way is now called Viremorpha cerani, so you may have to get used to hearing that. The Viremorpha apis we also measured, but it was almost always 0. It was very rarely seen, so we're not really reporting that. But for Viremorpha cerani, we found it in like 98% of the colonies and 88% of all samples, so it's pretty well universally present anyway. In terms of Vitiligenin, we saw no differences. That's not really a surprise because all of these bees were in the same apiaries, so they had the same forage available to them, and were able to build up the Vitiligenin levels, which you may know is higher in winter bees than summer bees. And this was winter bees that we were testing, right? So October bees. So they all kind of had the same amount of nutrition. Then we looked at some immune genes, and I'm more of a field person, but the lab people were super awesome. I hope you saw all of the authors on the first page. I should have named each one of them. But we had a graduate student, Brooke Lawrence, and postdoc involved in this laboratory stuff. And so defensin 1 is, as it sounds, it's like a defense reaction of the immune system. And it was more active in chemical-free bees than in the ones in the conventional and organic systems. Hyman optation is a different immune pathway, again, more active in chemical-free management system. And naked cuticle gene, also more active in the chemical-free system. So these all kind of went together to impact the survival of the colonies. So remember, we started with 288 colonies, so that's 96 in each treatment. And what you can see here is if you follow those colonies that were put in place in 2018, how many of them were alive over time. And you can see that the conventional and organic did much better than the chemical-free. In fact, out of 96 colonies without any treatments over 3 years, we had 1 remaining at the end. So it's not surprising when you look at the winter survival, which averaged about 27% for chemical-free. So when that happens year after year after year, the number of colonies really diminishes. So this is a crazy, pretty exciting figure called a path analysis. And this is trying to put everything together to figure out what is impacting what, right? So what we have here is if colonies were in the conventional or organic system, so the ones where we treated when mites got over the threshold, versus chemical-free, you can explain 78% of all survival, right? So it's management system matters is really the take-home message here. And then let's look at these arrows. So we have red arrows, which mean there was a negative impact, and we have yellow or orange that means a positive impact. And the amount of impact is based on the thickness, right? So very clearly, the management system had a huge negative impact on the varroa mite population, right? So treating for mites killed mites. Wow, surprise. There was also something of a negative impact on the amount of Nozema. There was a negative impact on the amount of DWV, negative impact on the expression of the genes that are defenses in the immune system, all three of these. And there was a tiny bit of an effect on Vitiligenin, although we know that wasn't significant because we saw that previously. Okay, so then let's go from the middle to the right. How do these measurements impact survival? Having mites had a negative impact on survival. So did having varroa, that V, I just want to say varroa, viremorpha. Deformed wing virus, we know, impacts survival, and the immune pathway with Hyman optation and naked cuticle gene had a negative impact on survival. But strangely, defensive 1 had the opposite effect. And not surprisingly, having more Vitiligenin, you were more likely to survive because that is a marker of health and nutrition. So this is kind of like, you know, you can take each of those graphs individually and see things, but really, this is a holistic look at how the bees were doing. How was their health? You know, did they have a lot of parasites, viruses, and was their immune system, you know, activated? So in summary, management matters. If the bees cannot control the mites, a beekeeper must do it for them. If you want to have colonies that are healthy, productive, and survive the winter. It's clear that controlling viruses is controlling varroa is going to control viruses. That's been shown many times. But really thinking about how it's also impacting the immune system and everything else. So I now think that the organic management system is best. So of course, controlling varroa mites makes sense. Everybody knows that that is going to help, but think about it. You don't need to use those synthetics. We had plenty of mite control and overwintering success without the synthetics. I can't tell you how many times I've heard you have to use amitraz. Well, you do not. You can use oxalic acid, formic acid, and thymol in rotation. You can use cultural controls, mite-resistant bees, screen bottom boards, all these other things. There's many, many options for you to use that are not synthetics. And now, this week, I'm hearing more and more that there are lots of mites resistant to amitraz anyway. So let's just stop and consider whether we can all have an organic management system. Now, all I mean by that is that you have a certain philosophy and you're avoiding synthetics. I am not saying that's all you need to do to get certified organic, because that's a whole entire other talk. So I just want to thank everyone that helped me. Clearly, I couldn't do this by myself. There was an army of people, lots of great beekeepers who came out to help me on a volunteer basis or for pay. And I know we're going to have lots of time for questions, but if you would like to send me a personal email, I'm super excited about my new special alias email, beekeeping@psu.edu. It's really fun to say. And I also want to talk about our next steps. So we call that the Comb Project, and we're just about to embark on Comb Part 2. So it took us 4 tries to get the funding, which, by the way, that project I just described was from the USDA NIFA OREI program. We applied and finally got the money from that program again to continue the work. And so what we're going to be doing now is using an organic management system for all of our colonies. And now we're going to be looking at how the bees use the landscape around them. So in order to actually get certified as an organic beekeeping or organic honey production entity, there's a huge importance on the landscape and the land around you and what's happening with it, right? And the current recommendations are you need a 3-kilometer forage radius and a 3.4 kilometer outside of that radius where there's nothing dangerous. And that's all based on how far bees can fly when they're desperate. But we know from Maggie Couvillon's work with dance language, she's from Virginia Tech, that bees, many reports are showing bees stay within about 0.7 kilometers. So we're going to place colonies on organic farms. We're going to have 2 management systems, both organic, but one's going to be single brood chamber management, and the other one's going to be double. We're going to do lots of landscape analyses. We're going to have do some radar tracking of individual bees to see where they're going. And we're going to have constant QR code tracking to see how long the bees are gone from the hive, which is a proxy for distance. At least we think so. Maggie Couvillon is going to be a partner in this, and she's going to have QR code bees and decoded dances and make sure that our technology is matching reality. We're also collaborating with Cornell University, which is Scott McArt's lab, where he does lots of pesticide analysis. So, we're going to place the bees on organic farms and hope that they are bringing back really clean products. So we're going to try to kind of close the loop on all the pieces of organic. We hope, maybe, or maybe the bees are going to fly too far and they'll just stay the same. But we need to look. I always, show me the data. I want to see why I need a 3-kilometer forage radius for organic. So anyway, with that, I'll take questions, and I want you to keep in mind that if you're interested in the year-by-year data, I did include those graphs in case you want to ask for them, and I can show them to you. Okay, so that was a lot in a short amount of time, and I was talking really fast, so sorry, but I will take questions. I'd love to hear them.
Great. So I'll open it up to the panelists first. If anyone has questions, don't be shy, and then I'll go through the questions in the QA.
Sure. I have a quick question. What was your funding level for the second part of your study? Approximate.
The study I just described to you was $1 million and 3 years. The second is $1.5 million and 4 years. Research is very, very expensive.
Yes.
And do you know for the second one, we don't have any money for equipment. It's ridiculous. So yeah.
Yeah, Robyn, this is Ron Miksha. I just wanted to say that I don't really have any specific questions at the moment, but I thought it was an awesome presentation. I really like how you built your case, and the final figure of the effect of management contributing maybe 75 or 80% of the final results in survival was very impressive. And you made a really strong case for organic beekeeping too. So it was really well done. I appreciate it.
Good, thank you.
Good. So just on the Q&A, so there's one question from your past. It says, did you treat a lot of bees at one time with formic indoors? So when you did formic indoors, what was your process, I guess? And were you doing 100 colonies at a time or pockets of them?
Yeah, I mean, I was sort of just trying it out, so we made mini overwintering buildings inside of our larger overwintering buildings so we could have the different treatments going at the same time. So I would do like 24 each, but I know that since then, at least in Manitoba, people have been doing it on a large scale. Yeah, the trouble with is I had to measure the acid on the outside and the inside of the hive, and it really did not want to go in. And I'm not an engineer, so I can't explain why that happens. But it went in enough to make a difference in mite control. And since then, further grad students have looked at just changing like CO2 levels and stuff like that. So that's pretty cool. And they've been able to harm varroa by the bees' own CO2, which I think is, you should look that up.
Yep. Good. Julia, did you have a question?
I was kind of curious about if you had any brood diseases, and if you did have brood diseases, how would you be treating them? And that's just because we have so much EFB up here in British Columbia.
Yeah, I actually teach a whole thing about organic management and not just varroa. But for EFB, my recommendations are to change the nurse to larva ratio, because EFB is really a stress disease and the larvae aren't being fed well enough. So either remove larvae or add nurses so that they can be better fed, and definitely feed them, right? So they can have as much food as they need. But that is what I recommend.
Yeah, that interesting. So you're the sort of fourth person now who's really focusing in on this as a way to change the demographic in there so they can look after the babies through the stress. So
Exactly.
yeah, I've started doing that in my blueberry pollination work. So thank you for that answer.
Sure. Yeah, the hard thing is having to get rid of get rid of larvae feels bad, right?
Yeah. Yeah.
It's not like you're giving them to another colony. You definitely don't want to do that.
No, and it's of course so difficult that I only have like 40 colonies in blueberries as opposed to thousands. So it's this interesting thing of what to do for the commercial guys who are having these problems too. So thank you. That's a bigger answer.
Yeah.
Thanks.
There's a question from Joanne around OA treatment amounts. So treatment amounts, I'm assuming grams, number of repeats.
Yeah, I feel like that's
Ju-
ever changing. Here in I just learned that New York and Delaware got some sort of registration for the glycerol treatment. Pennsylvania, we can't do that yet, so I can't recommend it. But yeah, so to me, one treatment with OA was 3 applications at a 5 to 7 day interval. And the way I worked it is I would have it with me when we were taking the varroamite alcohol washes, right? And so right in place, I could automatically dribble with other people around, right? And then when I would return for the second and third, I would just go by myself and do the vapor with my mask and everything like that, because I didn't have to open hives or anything, and I would be safe, and I would just vapor 2 more times.
Okay, were you doing 1
So-
gram per box? As per the label, or?
Yes, I always follow the label because I work for the University and Extension.
Sounds good.
None of this extra spicy stuff for me.
So I guess somebody was asking to see the intro slide again, but we'll keep that for last. So
Okay.
just you can bring it up if you like, and then I'll just keep going through the questions.
Hey,
So-
Dan, this is
yes.
Dewey. I have a question for Robyn too.
Go ahead.
Yes.
Robyn, in the presentation, you started the first year with all the colonies making their own comb, but you presented the overall measure for honey production. Were some of the details lost in summing everything together? Were there differences in terms of that first year compared with subsequent years in that aspect, and also in the aspect of, for example, of the Serranii infestations?
So here's my first slide. I don't know if people wanted to see the other authors, and then I will show you the data for that. So if I can quickly go down to the last few slides. So in terms of honey production, here you can see, of course, the first year it was almost non-existent. And then the second and third year were much better. And this is per apiary. So you can imagine, there weren't very many chemical-free colonies left. So that's sort of a big part of why they weren't making a lot of honey, right? Because they were dead. Not that it was funny, because it was really horrible watching them die. And that brings up a really good point. In the spirit of organic, you cannot refrain from doing something when there's a problem, right? So treatment-free beekeeping is not organic because you're not considering animal welfare. The only way it would be is if you are actually monitoring for mites. And if you see a problem, you're maybe requeening because you're a breeder or something like that. But to do nothing is not allowed. I had to do it for research here, which is horrible. But anyway, so that's a year-to-year honey production. Here's the mites. You already pretty much saw that in the line graph. And most of the stuff showed the same trends year after year. So it was kind of nice to see that. A lot of studies go for 10 months, 1 year. We followed the same colonies over 3 years. But I'm sure you're thinking in your head, okay, colony in a hive, it was not the same queen. Like clearly they swarmed and stuff like that over that and requeened and superseded and did all those things like they do for you. So, but bees in that box continuously for 3 total years.
Thanks.
get-
Thanks for the clarification. Appreciate it.
Yep.
I guess you sort of answered the next question around over the multi-year study. How did you handle swarm prevention? How did you consider split hives and hives that requeen themselves? Or was that just amalgamated altogether?
I didn't really answer that question. So the data that I presented here is what went into the publication, and the clearest thing was to follow the colonies only the colonies that were continuous over time, right? However, in reality, we did make splits to fill in the vacancies. And in particular, I wanted to always have 12 colonies in every apiary if I possibly could, because I felt like that density was going to matter for honey production and things like that. So sometimes the apiary started off with 4, 4, and 4 and ended up with 6 conventional and 6 organic, right? Because chemical free died, there weren't enough. I couldn't even split to get them back. I didn't buy any new bees. I didn't buy any queens. So all my splits were walkaway splits. And often that was my swarm prevention, right? So I would be like, okay, you're making queen cells. I'm taking the queen and, some stuff and moving it to a new hive and then leaving behind the queen cells so that they could requeen. And sometimes that ended up in the same apiary, sometimes the next apiary on the same farm, sometimes that farm was full, so they went to another farm. So they weren't included because that's like really convoluted and hard to talk about in scientific writing. Like it's not very straightforward. So following those colonies is what the paper will be about.
Okay, great. Next one is, this was ever so helpful and clear. Do you accept volunteers?
I do. You want to come to Pennsylvania?
So it's Debbie Blake, so she could probably email you if she's in Pennsylvania.
Sure. Yeah, you definitely want to come and see when we put little radar transponders on the bees and watch them fly. It's going to be so fun. And thank God we have engineers on our team.
Perfect.
Yeah.
Did you see any difference in the drone population between the treatment types?
I did not measure that, but what I found interesting was that the organic was the one where we were doing drone brood removal, and it definitely didn't impact honey production. My drone brood removal method was to actually, like, we had those green frames in, and when it was capped, we just scraped it off and threw it back in, because there was no going into the freezer back and forth stuff possibly going to happen. So we just scraped it off, which meant they had to redraw. So I thought it was going to impact honey production, but it doesn't seem to have.
Good. And I guess one question is, if you noticed EFB or AFB, how would you have managed that within the experiment?
Yes, AFB, we would have had to, of course, burn the colony. EFB, I already kind of went over how we would try to influence the nurse to larva ratio. We definitely saw that. We saw sac brood like a lot, did nothing about it. We sadly saw some pretty bad PMS, DWV, deformed wings, horrible in the chemical free. Weren't allowed to do anything about it. Yeah, so when you keep bees as a researcher and each colony is its own experimental unit, there's like a lot of beekeeping stuff that you can't do, right? Like we did not combine colonies for winter or anything like that because each was its own unit. So we had to just, if it was a small cluster, good luck. I'll see you in spring. You know?
There's a quick question on formic acid. So it's probably a label thing, but it says formic acid 90%. How much water would you mix to get the best results? What would you dilute it to?
We,
Or
here
are you
in the
just using
US,
Formic Pro?
yeah, there's no formic pour-on treatment allowed where I am. So we only use Mite Away Correct strips or Formic Pro.
Mhm.
If you're talking about in the wintering building, it sort of depends on what dose you want, what percent is going to be in the liquid. So, which is also not registered here.
Yeah. Let's see. Are there any studies or data on Apidology versus other organics? Have you ever heard about that?
Yeah, so that Apilife var, that's like a thymol eucalyptus wafer. I think that the only component that's mentioned in the organic standards is thymol, so I would not think that that would be technically for certification probably currently allowed. But again, you're not going to have the land anyway, so I'm all for it. And if I can, my dream one day is to actually influence the standards when they rewrite them soon, because they're from 2010, so it's about time to renew them, I would think. If they ask my opinion, I would be very happy.
The next one's a loaded question, so I'll skip it for now.
Thank you.
Because it'll take a while to answer, because it's about re-explaining. We are recording this, James, so you'll be able to re-watch the section that you need, where she actually explains the arrows and the negative and positive impact. So, because it was quite the impactful slide. I really liked it. I'm an engineer. I love data, but it's recorded. It'll be posted in the next few days. The next one,
And maybe published soon with an explanation, maybe.
Nice.
Yeah.
On your new project, how are you doing QR code tracking? Is it just using technology like cameras and stuff?
Yes. So the computer attached to a camera, and the computer is like a little Raspberry Pi unit, and they come with this little camera that's going to be pointed down at the entrance. And the engineering students are working on software to, integrate the QR codes. Have directionality, so you can tell whether they're coming in or going out based on, as long as we put them on them properly. And each bee will have its own like number. So yeah, whenever the camera detects there's a QR code, it's gonna like catch a photo and then convert it to a number and put it in a spreadsheet, which sounds very straightforward, doesn't it? Well, it's gonna take them, it's not that easy. I mean, engineers, they can do anything. It just takes a while to get from concept to reality. But there's a guy at Cornell University, Nils Knapp, N-A-P-P, and he has published on doing some of this QR code work. And luckily he's nice and talks to us and
Good
is helping us.
We're almost through the questions. Were you able to convince any of the non-chemical beekeepers to change their methods of managing?
I did convince one, and the best thing was that I hired him to actually help us manage the colonies. And he had been a treatment-free beekeeper, but then he kind of realized that he should be monitoring for mites, to say the least. And I also was excited to see him want to collect data and like understood the importance of tracking hives and things like that. So now he kind of promotes the idea as well, you know, the animal welfare piece of it and the monitoring. I think so earlier tonight, I listened to someone else speaking. It was Troy Hall from New Hampshire talking about coming back from the brink as a treatment-free beekeeper, lost 90% of his colonies. And he now does mite washes as well. Like, I think once you start and you start to see the level of mites, whether they're super low because you should be breeding from this colony or they're getting really high, I think the reality is there for you as a beekeeper to see what your bees are dealing with. So
That's perfect.